Scott Howell, MD, PhD, from the Research Center at Tier 1 Health and Wellness in Chattanooga, Tennessee. So he's with us. Most of you guys will know him from our Facebook group, TRT and Hormone Optimization Therapy, HOT. And today Scott is with us to talk and explain all about the relationship between the androgen receptor and downregulation and expression of the androgen receptor.
Okay, I'm going to start now. Thanks, Stephen, for having me on. This is a topic I've been very passionate about because so many people have asked in the Facebook group and in general whether testosterone, increasing testosterone dose increases or actually downregulates the androgen receptors. This has been a never-ending topic for most of my adult life. Several well-known TRT practitioners and some naturopaths here recently have made this claim on YouTube. And one physician that used to be in the Facebook group used the term burnout to imply that higher testosterone doses desensitize androgen receptors, making them somehow less effective. Now, on the surface, this seems like it would be a reasonable claim, but the problem here is that there is a fundamental ignorance of the basic endocrinology and physiology when it comes to sex hormone metabolism. In the 1980s, this was the prevailing view in the scientific literature. And even in 1996, Bill Phillips published the World Anabolic Review. And in this book, he made this claim as well, that increasing a dose of androgen would downregulate the androgen receptors, although there was compelling evidence at this time that countered this view. Somewhere along the way, some clinicians and patients have equated a downregulation of androgen receptors to the phenomena of tolerance. I just discussed this this week with Dr. Keith Nichols, and I can assure you, all of our audience members, that this is simply not true. Viewing this issue of downregulation from a tolerance, saturation, absorption, or sensitivity perspective does not come anywhere near close to explaining what actually happens. So like Stephen mentioned, the questions to be answered in this podcast include: what is the relationship between androgen receptors and negative feedback? What regulates androgen receptor expression? And by characterizing these two questions, we will fully answer the last question of whether increasing testosterone dose downregulates the androgen receptors. So everyone should know the history of testosterone. I'm really big on going into the historical context. The majority of you probably know that the first synthesis of testosterone occurred in 1935, but most of you probably don't know that one of the first two synthesis processes at this time was a methylation synthesis to derive methyl testosterone from cholesterol, when Ruzicka attempted to mimic the natural synthesis pathways in the body. Now, although Ruzicka published his synthesis 1 week after Butanant, his synthesis of methyl testosterone allowed oral absorption to bypass second pass metabolism without intending to do so at the beginning of the history of androgens, synthetic androgens. This is why testosterone is not an anabolic steroid. It was after the initial 17-alpha-alkylated synthesis that researchers began to try to dissociate the androgenic effects from the anabolic effects. And many scientific papers of authors who should know better have applied and reported a post hoc conception of anabolic steroids to testosterone, and this is simply not correct. Testosterone was and continues to be the yardstick by which anabolic steroids and SARMs are measured by. So the errors in historical context go back even further. Ruzicka and Butanant did not specifically receive Nobel Prizes for their synthesis of testosterone, like what has been published in many papers. Butanant received the Nobel for his work to identify the molecular structure of sex hormones that included testosterone, and Ruzicka received the Nobel Prize for his use of polymethylines and higher terpenes in not only the synthesis of testosterone, but also in the synthesis of perfumes as well. So you are probably thinking or asking yourself right now, why does this matter to androgen receptor downregulation? Well, from 1935 to the present day, 84 years have elapsed, and androgens became one of the most extensively studied drug classes in existence. So the answers to many of the questions that are posed in the forums and the questions we are answering right now have been known in the literature for quite a long time, but no one goes to the length to dig it up and go through the literature. So now we turn our attention to negative feedback loops. The issue of equating androgens to the conception of tolerance is a fundamental ignorance of the basic homeostatic model on this slide and how tolerance actually works in the body. For many physiologic processes, like the regulation of body temperature, the model fits very well. A receptor senses a change, the receptor signals to a control center, and then the control center initiates an action in an effector to decrease or increase a physiologic parameter. So essentially, we have three components of negative feedback: a receptor, or sensor, a control center, and an effector. Each of these components works to keep a hormone or physiologic process in homeostasis within a certain range, conducible to maintain life. So now we look at the HPG axis. The hypothalamus pituitary gonadal axis has 4 points in which negative feedback occurs, similar to the homeostatic model on the previous slide. The kisspeptin system acts as a sensor. The hypothalamus acts as the control center. The gonadotropins function as effectors, and the Leydig cells function as effector cells. The set point for testosterone is very important because set points for physiologic parameters can change given different circumstances, such as an infection. With the infection analogy, the body compensates by increasing the body temperature, and this is why you run a fever, and when the fever breaks, the set point resets to a lower value. Now, depending on the set point, peripheral levels of testosterone, other sex hormones such as estrogen and progesterone, along with metabolic signals like leptin, are sensed by the kisspeptin system in the arcuate nucleus of the hypothalamus, which is the control center. And this stimulates KISS1 expression. And this in turn stimulates GnRH neurons to release gonadotropin-releasing hormone into the portal pituitary circulation. Then GnRH binds to its receptors in the pituitary, stimulating the secretion of the gonadotropins into systemic circulation. The gonadotropins include the effectors known as luteinizing hormone, or LH primarily, and follicle stimulating hormone, or FSH, to a lesser extent. Luteinizing hormone then stimulates the Leydig cells in the gonads to produce testosterone. So ask yourself this question: where are androgen receptors controlled in this negative feedback? Although androgens stimulate the kisspeptin system, other sex steroids and metabolic hormones also stimulate the system. Androgen receptor binding acts only through the androgen receptor DNA binding domain to repress GnRH transcription as one of the many regulators of GnRH release. The entire process has nothing to do with androgen receptor expression in target tissues. Androgen receptors are not controlled by negative feedback, and for good reason that we'll see in the upcoming slides. So before we jump into how androgen receptors are regulated, it is good for us to visualize what we are talking about and to see how complex the androgen receptor and its functions are. The androgen receptor is a protein that's made up of 919 amino acids in healthy people, but with some polymorphisms, the number can be as low as 910. The androgen receptor contains a ligand binding domain, AF2, at the top right in green, a DNA binding domain at the top center in orange, and an amino terminal domain, AF1, at the top left in cream color. So obviously here, the natural hormone agonists of the androgen receptor are testosterone and the 5-alpha reduced metabolite, dihydrotestosterone, or DHT. So now we look at the classical model of androgen receptor action. This is the model that's taught in medical school and presented in most physiology texts. In the absence of androgens, the androgen receptor is located in the cytosol as part of a multi-protein complex with several chaperones that are essentially heat shock proteins. And there's a co-chaperone, P51, as well. When either testosterone or DHT diffuses through the plasma membrane and binds up here where the membrane is, it comes in and binds, this causes conformational changes in the androgen receptor protein complex. And this binding permits the dimerization or adducting, this contracts inward, of the androgen receptor and what is referred to as the NC-terminal interaction right here. Then the complex translocates to the nucleus where receptor dimers are stabilized and bonding to DNA disrupts the NC-terminal interaction. This opens up the rooms of the AR terminal domain for interactions with the cellular transcriptional machinery. The machinery includes many co-regulators that recruit RNA polymerase to create messenger RNA, or mRNA, that eventually gets shuttled back to the cytosol to undergo translation at ribosomes to produce functional proteins. So the important point with including this slide is that the process from testosterone diffusing through the plasma membrane, binding, translocating to the nucleus, and then producing functional proteins in the cytoplasm takes hours to demonstrate a physiologic response. Excuse me. Non-classical androgen receptor action works considerably faster than the classical model. To understand this graphic, it is helpful to know that because the androgen receptor functions as a transcription factor, our molecular understanding of the role of testosterone has been established through the identification of androgen receptor target genes. These genes were identified due to the presence of androgen response elements in promoter regions. Due to all these steps, changes in gene transcription can take several hours to show a physiologic response. However, in 2004, isolated Sertoli cells were shown to rapidly respond to testosterone stimulation in less than 1 minute by upregulating the activity of MAP kinase signaling, which was found to be dependent upon the androgen receptor. So we have two signaling pathways. We have the classical that takes several hours, and then we have a non-classical that is still dependent upon the androgen receptor that occurs in seconds. The seminal study by Tuchek 2016 used specific activators and inhibitors in both the classical and non-classical signaling to show that both pathways shared overlapping functions that retain distinct roles in spermatogenesis in the Sertoli cells. In non-classical AR signaling, testosterone stimulation transiently increases androgen receptor localization. And what that means is that the androgen receptors in the cytosol move to the plasma membrane. This results in the localized androgen receptor activating SRC tyrosine kinase, and that is here. This activation in turn alters phosphorylation, you see where the P's are, and activation of the EGF receptor here. That additionally activates the MAP kinase cascade that includes RAS, RAF, and ERK. Further signaling through the P90 kinase pathway causes phosphorylation of the Krebs transcription factor, here resulting in increased transcription of Krebs-inducible genes. Now, armed with our knowledge of both slow and fast androgen receptor actions, we look at the evidence of how androgen receptors are regulated. I will use the term upregulation, but in reality, androgen receptors are auto-regulated or self-regulated at the tissue level of control. And this has very important clinical implications when we talk about dose response. So even before the research that is shown on this slide, there was a solid amount of literature that basically hypothesized that there would be downregulation, but in the actual published write-ups, there was evidence of upregulation. Many of the researchers believed at this time that downregulation would occur and attempted to point to various mechanisms for the contradictory results that did not meet their a priori hypotheses. So here, Gadd in 1988 observed upregulation of androgen receptors in genital skin fibroblasts upon prolonged exposure to DHT and an anabolic steroid called metribolone. Now, metribolone is an oral form of trenbolone that is 17-alpha alkylated and was referred to at this time as Research Chemical 1881. At 20 hours of exposure to both of these hormones, there was a 2.07-fold increase in upregulation by DHT and a 2.35-fold increase upon exposure with metribolone. The authors concluded that upregulation plays a role in the high affinity binding of androgen receptors in the nuclear matrix. So here, Gonzalez, 1993, looked at smooth muscle cells cultured from immature and adult rat penises to assess downregulators of androgen receptor expression as a function of cell proliferation and smooth muscle of androgen responsive vascular tissue. So here, proliferation means just to increase the number of a specific cell type. And androgen responsive vascular tissue just means that there's androgen receptors here and that they can bind. So what the researchers did was apply metribolone to each of the cell types, or to each cell type. They found that contrary to their hypothesis that androgen receptors would be downregulated, they were actually upregulated in adult penile tissue. When they prevented contact and depleted the serum of androgens, this downregulated androgen receptor expression, but upon re-exposure with DHT, the downregulation was countered. Excuse me. So here we have the first inkling of a dose response. So here, Dumit in 1996 investigated whether androgen receptors existed in skeletal muscle satellite cells and myotubes, and they actually looked at the effect of testosterone on satellite cell proliferation and differentiation. So again, proliferation just means to expand one cell type. And differentiation here means that the cell has options. And in this case, the cell can go into an adipogenic lineage or fat cell lineage, or a myogenic lineage into a muscle cell. So the researchers found that in both satellite cells and myotubes, AR increased at each measurement increment at 6, 12, and 24 hours of testosterone exposure. The authors concluded that satellite cells were direct targets of androgen action, and testosterone administration increased the numbers of androgen receptors. So this study by Wyron and researchers looked at exposure to DHT and AR expression in human osteoblasts. Specifically, they looked at two cell types, SAOS-2 and U2 bone cells. So they found that DHT increased AR expression in a time and dose-dependent manner. And interestingly, when they applied an anti-androgen to these cells, it attenuated the upregulation. The authors here concluded that physiologic concentrations of androgens had a substantive effect on the expression of androgen receptors in bone tissue. Now, this study by Lynch assessed upregulation of androgen receptors with several anabolic steroids and DHT. They injected 2 milligrams per kilogram of testosterone cypionate or nandrolone decanoate, 1 milligram per kilogram of boldenone, or 5 milligram per kilogram of DHT in castrated rats. After 18 to 24 hours of exposure, they sacrificed the rats and excised the brain tissue. They found that each steroid type upregulated androgen receptor expression in the ventral medial hypothalamus, the medial amygdala, and the medial preoptic area. So far, we have covered a diverse amount of tissue. We have penile tissue, both fibroblast and vascular. We have brain tissue now, and we have muscle tissue where upregulation has occurred. So this study by Hankey looked at the mechanisms behind the protective cardiovascular effects of testosterone in rabbits. And they found that androgen receptors were upregulated, and this occurred in major vascular tissue. Not only did testosterone inhibit new enamel plaque development, but it also increased AR expression by 50% in each of the arterial segments that was assessed in this study. So despite the claims that nitric oxide release is the primary cardiovascular benefit of testosterone, I argue that direct androgen receptor action is the primary benefit of optimization therapy in androgen responsive vascular tissue. Specifically, androgen receptor action to prevent new enamel plaque development, action to reduce adhesion molecule expression, action to decrease inflammatory cytokine production, action to favorably modulate the lipid profile, and to increase vascular reactivity. And finally, to mediate nitric oxide release. These are the primary cardiovascular benefits of testosterone that no one really talks about. So this study looked at vascular tissue, specifically aortic vascular smooth muscle cells of rats. Here, the researchers characterized the time response from 0 to 48 hours of testosterone exposure. They found androgen treatment for just 10 minutes induced a transient downregulation of cytoplasmic androgen receptors, but afterwards there was a gradual increase in AR expression that plateaued at a 5-fold increase at 48 hours from baseline. Nuclear androgen receptor increases became evident at 24 hours, but reached greater AR expression at 48 hours. So the researchers concluded that androgen exposure to physiologic concentrations increased AR expression in vascular smooth muscle cells at the level of transcription and non-transcription. So moving back to the slides we covered previously, the classical model and the non-classical model. So here we have evidence that this is occurring with vascular smooth muscle cells. This reconfirms the mechanisms for the protective cardiovascular effects we mentioned on the previous slide, that direct AR action coupled with fast signaling to increase vascular reactivity, prevent new enamel plaque development, to lower inflammation, to modulate the lipid profile, and to stimulate the release of nitric oxide. So this study by Carson and researchers looked at androgen receptor expression with high-dose nandrolone in rat hind limbs. They used 6 milligrams per kilogram, which equates to roughly 545 milligrams of nandrolone per week in a 200-pound man. They found in the soleus muscle that nandrolone increased androgen receptor expression by 250% in 5-month-old rats and 900% in 25-month-old rats compared to control rats of the same age. So here, nandrolone increased androgen receptor expression with increasing age, likely as a compensatory mechanism. So we see an interesting finding here of an effect modification by nandrolone with age to increase androgen receptor expression. So this study by the Basine group demonstrated that androgen receptors were upregulated with both DHT and testosterone in mesenchymal pluripotent stem cells of mice. These somatic stem cells have two options for differentiation. They can either take a myogenic lineage into muscle cells or into an adipogenic lineage for fat cells. So here the researchers not only provided more evidence of upregulation, but also showed that DHT and testosterone regulate stem cell lineage determination by promoting these mesenchymal pluripotent stem cells to differentiate into muscle cells instead of adipose tissue cells. This is another unmentioned positive effect of testosterone replacement therapy that no one talks about, and it explains in part how the body recomposition of patients occurs in those that are under androgen therapy. So now we move from the animal studies to the human studies. And here, Cotty and researchers assess 8 high-level powerlifters without anabolic steroid use, 9 high-level powerlifters with anabolic steroid use, and 6 untrained and unusing subjects as controls. So the intent here was to determine, excuse me, whether long-term strength training and self-administration of anabolic steroids independently and together acted to change androgen receptor content in certain muscle types. So we're not looking at cells anymore, we're looking at the expression in muscles. So after biopsy in the trapezius muscle, they found AR-containing myonuclei were higher in non-using powerlifters compared to controls, but they also found the anabolic steroid users had higher AR content than both non-using powerlifters or controls. In the vastus lateralis, the myonuclei number was higher in the anabolic steroid using powerlifters compared to non-using powerlifters and controls. So they concluded that androgen receptor increases differed by muscle type according to strength training and anabolic steroid use independently of one another. And this is another line of evidence showing autoregulation given different muscle types at the tissue level of control. So now tying everything together that we have looked at so far to this point, this study by the Basine group in 2004 assessed biopsy samples of 6 healthy men that were administered 600 milligrams per week of testosterone and anthate for 20 weeks. Most any of the professionals that are listening to this podcast right now know these Basine studies. Now, building on the study presented in the previous slide, the researchers sought to determine which muscle tissue cells, rather than muscle type, expressed upregulated androgen receptors. They found that AR was expressed predominantly in muscle satellite cells, myonuclei and myofibers, CD4 stem cells in the interstitium, fibroblasts, and mast cells also expressed higher androgen receptor content. They also found higher androgen receptor expression in endothelial and smooth muscle cells and concluded that high-dose testosterone increased androgen receptor expression in satellite cells and myonuclei while increasing nucleolar androgen receptor density. I included this slide to demonstrate how science sometimes proceeds in a backward fashion in the academic literature, and how researchers must change their beliefs in the face of evidence. We just saw on the previous slide that Basine's research group found greater androgen receptor expression in one of the high-dose testosterone studies that used 600 milligrams per week in healthy men back in, the study was in 2004. Now you will see what the very same research group claimed in 2001 when the actual trial, one of the actual trials was conducted. Here, 3 years before they found higher androgen receptor expression in many muscle cell types, they stated verbatim in this write-up, androgen receptors in most tissues are either saturated or downregulated at physiological concentrations. They cited 3 animal studies and 1 review as evidence for this claim. However, when I read this, I was really disappointed to see a primary piece of research reference a literature review as evidence to support this claim. We have established in this short podcast that the evidence during that time in 2001 did not warrant this claim. And it should be clear by now to anyone that's viewing this podcast that any instance of saturation would be transient, because autoregulation by the androgen receptor gene shifts continuously in a responsive manner at the tissue level of control. This means that for saturation to occur, it would have to do so in many target tissues simultaneously, which we know does not happen based on the evidence we have seen so far. This is why the word saturation to describe androgen receptor binding is almost never used in the correct context and has no real meaning to characterize the general process of androgen receptor binding. Basine and his group changed their belief. I decided to include this slide to show the clinical importance of autoregulation in other fields of medicine outside of testosterone replacement therapy. Specifically, how errors in autoregulation result in paradoxical effects of androgen receptor expression in castration-resistant prostate cancer. Here, instead of downregulating androgen receptor expression in response to castration levels of androgens, as would be the case in normal conditions and in healthy adults, castration-resistant prostate cancer cells adaptively autoregulate AR expression by increasing androgen receptor content to 25-fold or greater in lower androgen environments. So in this study, upon exposure with metribolone, trenbolone, the methylated trenbolone drug, upon exposure to this, prostate cancer cells did not proliferate as seen with controls, or to a lesser extent with anti-androgen exposure. Here, exposure to metribolone lowered prostate cancer cell number as shown in figure A, or attenuated cancer cell proliferation as shown in figure D. This paradox shows why bipolar androgen therapy is used to treat castration-resistant prostate cancer. Normally, androgen receptors would increase with increasing androgen dose, but here the exact opposite occurs. When androgen levels drop, the androgen receptor expression increases. So, to answer the first question we posed, what is the relationship between androgen receptors and negative feedback? Although endogenous androgen levels are controlled by negative feedback, androgen receptors are not controlled by this mechanism. Androgen receptor expression is autoregulated at the tissue level of control by the way of the AR gene. The AR DNA binding domain acts as a repressor of GnRH transcription as one of many other hormones that regulate the release of GnRH in the HPG axis. The HPG axis has nothing to do with the expression of androgen receptors in the target tissues. So what regulates androgen receptors? So far, we have seen that physiologic concentrations and high dose concentrations of testosterone, DHT, metribolone, and several other anabolic steroids upregulate androgen receptors in penile tissue, penile vascular tissue, arterial tissue, vascular smooth muscle cells, several brain tissue cells, satellite cells, and myofibers in muscle tissue, and at least 2 bone cell types. We see that the effect is dose responsive, meaning that as androgen levels decrease, the androgen receptor gene autoregulates by decreasing the androgen receptor number. We have seen how errors in autoregulation are involved with castration-resistant prostate cancer, and we also see that as androgen dose increases, the androgen receptor gene autoregulates by increasing androgen receptor expression. This is the primary reason that extreme anabolic steroid doses continue to work and be effective because the body compensates for this increase in androgen load by stimulating the androgen receptor gene to produce greater numbers of androgen receptors in the target tissues. Androgen receptor expression is controlled by the process of autoregulation independent of negative feedback. All sex steroid receptors have some degree of evidence of control by autoregulation. So contrary to recent claims, increasing androgen dose to any level does not burn out or downregulate androgen receptors. So finally, we answered definitively whether androgen receptors are downregulated with increasing testosterone dose, and the answer is a resounding no. This has been known for at least 30 years, and I've traced it back even further than that. Now, one final note before I take any questions, and I apologize it's taken so long. I tried to move pretty fast. For any practitioners that have fell into this claim and have been taught faulty information or picked it up somewhere along the line, Dr. Keith Nichols, Angie Nichols, and I will be offering intensive physician training on-site and remote with several introductory and advanced courses on androgen metabolism and research methodology that you can register for at tier1hw.com. Our intent is to bring the field up to speed and to forge a path in evidence-based practice. If you would like to become a Tier 1 trained physician or allied health professional, you can contact me at Howell at tier1hw.com or call the practice at 931-520-0200. I look forward to hearing you. Look us up on Facebook, Twitter, and Instagram. Back to you.
Okay, thank you, Scott. That was very interesting. A lot of stuff to take in. So we have a question. When and how, if possible, will these findings be applied to patients? What can we do with that?
Could you repeat that?
Sure.
Are you taking this from the actual box
Yes.
online?
Yes, this is from the chat box online, yes.
Okay,
So,
I'm going to go there.
So the question was,
go ahead.
When and how will these findings be applied to patients?
Well, the issue of downregulation of androgen receptors is it's often used to justify the use of other drugs or other practices that aren't warranted. So there is no sensitivity with androgen dose unless a person has a pathology, whether they have a malfunction of the androgen receptor. And most of the time, this is genetic. So as far as being applied to practice, it's about disseminating the correct information. Patients should not be concerned about receptors burning out because it just doesn't occur. It doesn't happen. It's not supported by the literature in any shape or form.
Okay. Have there been some research on how far this upregulation of the androgen receptor will go? Will there be any upper limit? Has there been dose-dependent studies with anabolic steroid use?
The highest studies that I can recall in the literature are, of course, the Basine studies that used 600 milligrams per week. There's been other studies that have shown an upregulation with higher doses than that in conditions of abuse, but there was no comparisons. There was no controls to compare it to. I think everyone has to view this from a different perspective. The reason why androgen receptors are controlled by autoregulation is because the body has to have some type of mechanism to cope with fluctuating levels of androgens at any one given point in time. So if an individual have a higher testosterone level than normal, their body compensates by stimulating the androgen receptor gene to facilitate the bonding of those hormones. So as far as the upper limit, to be honest with you, I've never seen one in the literature.
Okay. Another question was this delay in upregulation of androgen receptors. Can this be the reason that patients starting on TRT, for example, need to be so patient? Because there needs to be time to upregulate them. And if so, how much time, Scott?
I really don't think that that upregulation is a big issue with this. I think that when people respond to testosterone therapy, that their sense of well-being and all that, it takes longer than what it does to actually have the physiologic response. So if once you initiate androgen therapy, there's about a 2 to 4 week window that most people experience where they see some type of changes, minor changes in body composition, but they have a fairly quick response to libido issues. So although the slow signaling pathway is involved, once the system is, once your testosterone levels are binding to androgen receptors in the tissue, the response just takes time, and there's no way of getting around that. Did that answer the question?
Yes, sure it did. Okay, thank you. That was all for the questions for now. Of course, viewers can still ask questions in our Facebook group, TRT and Hormone Optimization Therapy, where Dr. Keith Nichols and Dr. Scott Howell as well are members of and participating almost each day. So one more thing, Scott, because I could already see your slide number 27 there on the screen, androgen receptor regulation, the myth of blasting and cruising. You would announce further podcasts on this subject?
Yes, I have started to compile the information for this podcast, and there have been several gurus with anabolic steroids that have been talking about absorption, sensitivity, all these to justify this practice of blasting and cruising. And just a short primer, blasting is using really huge doses of androgens over a short period, usually from 2 to 4 weeks, and then cruising for 2 weeks on replacement doses. And so what we're going to cover here is the actual history of where blasting and cruising came from and why desensitization or downregulation does not justify the use of blasting and cruising.
Okay, that will be great. I am looking forward to that. So that's really the science behind the blasting and cruising. Okay, so rest me to thank you so much, Scott, for all the insightful information, for covering the research for us. I know that's what you like to do each and every day. A lot of research going on still.
Oh, yeah. Yeah, hopefully we'll be geared up for trials by next year, or maybe a little while right after that. But hopefully we're going to conduct a pharmacokinetic study of cream, the first study Keith and I are talking about. So we're going to get that knocked out so people have better data to look at.
Okay, thanks. Okay, so we'll end this podcast here. I'll talk to you next time, Scott. Thanks so much.
Thanks, Stephen. Take care, buddy.